This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on December 10th, 2019. I'm Vincent and you're listening to the podcast all about viruses. Today we are recording at the Nipah Virus International Conference in Singapore, which recognizes the discovery of this virus 20 years ago. And I've assembled a panel of individuals to give you a flavor of some of the key topics that have been addressed at this conference. My guest here, right across from me, he's from Duke NUS Medical School. He's also a co-organizer of the meeting, Linfa Wang. Welcome back.
Thank you.
And you were previously on TWiV 296.
That's right.
Which was called The Real Batman.
Yeah, thanks for that. You got me a lot of publicity for that.
Great. You gave me a beautiful little bat, too.
Yeah, that's right. Yeah.
Not a live one, though.
Yeah.
Also joining me here from Curtin University, John McKenzie. Welcome.
Thank you kindly.
My pleasure. Next to him for a second time from the EcoHealth Alliance, John Epstein. Welcome.
Thank you, Vincent.
You were on episode 501.
Almost got 500, but
which was Outbreak.
Yes.
Which we were so lucky to do right in the Smithsonian exhibit, right?
Mm-hmm. That was a lot of fun. Yeah, it was great to show you that exhibit.
Terrific. And finally, from the NIAID at the NIH, Unchung Park. Welcome. Hi. I have to tell you that Ian Lipkin made me practice saying your name a lot.
You did well.
That I did well?
Yeah. So he was a good teacher then.
Yeah. No, he's corrected me several times. And so he was very good at saying that.
Ian Wood. So
we're going to talk a little bit about very specific topics from this meeting, which all of you have addressed. So let's start by asking you, why do we have a Nipah virus meeting, Linfa?
For two reasons, I think. The first reason is that to mark the 20th anniversary of the discovery. And the second reason, I think, is really important for CEPI, the Coalition for Epidemic Preparedness Innovations, because it's really a novel concept.
Right.
And I always say for disease like Nipah, Ebola, I call them high impact, low frequency. These kind of disease, the countermeasures are difficult. The traditional development vaccine by farmers will not work. So that's the reason. And, CEPI approached me, and I was very glad to take the challenge and organize it in Singapore.
It's quite a challenge, right?
Yeah, it was a short time because I met, the CEO, Richard, in March, and then they had an internal discussion. The formal decision to host the meeting here was April. And we have to do it December 9th,
So that's how many people from how many countries do you have?
Yes, totally we're close to, originally I promised him to 130 because Nipah is a relatively small kind of niche.
Yeah.
But we, exceeded that by a big margin. So 250 final kind of participants from 24 different countries.
And how did you figure out what topics to...
That I rely on international organizing committee. So we got involved, very early on with the International Honor Committee to decide what's the topics and who are to invite.
Right. So was it a success, do you think, this meeting?
I'm biased. It's a huge success.
Okay. Will there be a Nipah 30?
Well, I don't know. I mean, I have mixed feelings. On one hand, if you can eradicate a disease, you don't need a Nipah 30. But I think the scientists, will be out of job. So yeah. I personally think there will be Nipah 30. As you say, with other disease, even for smallpox and things like that. I think you still have science going on even after successful control.
Still some polio going on.
Right. Yeah.
I don't think we'll ever be out of work. That's unfortunately true.
I thought yesterday, looking at all these people in this room, what would they be doing if there were no Nipah viruses?
There would be disease X, right?
Yeah,
so this was the
That's exactly right. They would do something else.
In fact, you mentioned polio. In fact, we have a polio emergency committee meeting tonight.
Really?
Yeah.
What's that about?
Well, it's the WHO emergency committee.
Yeah.
And looking at trying to finally get rid of the last few cases, but it's proving intractable.
Because the vaccine reverts and spreads.
That's right.
That's a problem.
That's right.
Yeah. So John, you talked at this meeting about a little bit of a historical overview. So maybe you could summarize for our listeners the history of Nipah virus as we know it.
I guess in that sense, it's probably better to talk about the history of Nipah viruses rather than Nipah only. And this really started in Brisbane, in Australia, in 1994. And when we had an outbreak in the stables where some racehorses and a racehorse trainer died from an unknown disease, found very quickly to be a paramyxovirus-like cause. It was called equine morbillivirus incorrectly. Finally, they realized once we had the sequence that in fact it was a quite different virus. The first member of a new genus, or the new member, shall we say, of the family. And so Nipah was the second one that came along 5 years later. And so we had two, I think, important aspects from the first outbreak, the Hendra outbreak. One was the fact that it was a new virus and a new genus. And the second was that the reservoir host were bats. And these were two very, very important aspects for when we came to the Nipah outbreak. Then we had the first big outbreak here in Malaysia and Singapore, where we had about 107 deaths and about 260 cases. We then had a slight break before it suddenly reappeared again in Bangladesh. And in Bangladesh, we've had a whole series of outbreaks over the years. Then two of them occurred actually in West Bengal, just across the border from Bangladesh. Siliguri, another one, that was the first one, another one about 7 years later. We had then the outbreak in the Philippines, which is a little bit strange. This was in horses and humans. So rather than having pigs, as it was in Malaysia, as the major intermediate host, we had horses, a bit more like the Hendra virus in a sense, but it was still Nipah. And most recently we had the outbreaks in India, in Kerala, somewhat unexpected.
So the original outbreak in Malaysia was first in a pig farm, right?
Correct. And it was initially it was thought to be due to, in terms of the animals, the pigs, thought to be probably classical swine fever.
Right.
But meanwhile, people were dying too. And one or two cases were diagnosed as being Japanese encephalitis, and the diagnosis, in fact, was confirmed by the reference laboratory up in Japan, in Nagasaki. But unfortunately, a number of things hadn't happened. They decided to vaccinate widely, and also to do a lot of fogging, insecticide fogging. People still continue to die despite that. And one of the big problems, I think, was there was no communication really between the veterinary and the human. If you like, this is the first good example of where One Health mechanisms failed. It didn't exist. And it's a major lesson we've learned from that, I think, is that we really do need a One Health approach to these zoonotic diseases.
Right.
So I think that it wasn't until Paul Chua isolated the virus for the first time in early March or late February that we realized it was a totally different virus. It was very quickly shown to be related to Hendra. And the rest is history, shall we say.
So I was reading some of his articles, and he said it was very easy to grow in cell culture, right?
I have to tell you that Paul Chua has green fingers. If there's anything around that'll grow, Paul will grow it. It's quite incredible.
Yeah, it's true.
And this took place in the Department of Microbiology at University of Malaysia. And yes, it was really a very important issue.
So at some point, virus was sent to Fort Collins because it was thought to be an arbovirus, right?
Paul Chua took it over to Fort Collins and on to Atlanta. Oddly enough, Ken Lamb, who was a professor of the department in which Chua was working, had phoned me up and had described the CPE, which is giant cells, Cecilia. I said, it sounds just like Hendra. So he then actually sent a message to both Dwayne Gubler in Fort Collins and also to Brian Mai in Atlanta to say that check out Hendra at the same time while you do the test.
So the formation of syncytia would not have happened with Japanese encephalitis, right?
No, absolutely not. Yeah.
Okay.
Yeah.
And then they used Antisera, I think, as well to confirm that.
They did in Atlanta, yeah.
Atlanta.
That's correct. anterior to Hendra.
So at that point, it moved out of Fort Collins because it wasn't an arbovirus. That was their specialty there at CDC
Correct.
Fort Collins, right?
But it had already been arranged that I think that Paul was going to go both to Fort Collins and to Atlanta.
Right.
So that had been pre-designed, shall we say.
So at what month of the year was CDC sure that this was another Hendra-like virus?
Beginning of March.
99?
99, yeah.
Okay.
At that stage, it had been going for about 5 months, the outbreak.
Okay. Now, if you read some of the review articles, people thought this was Japanese encephalitis. They were stunned when it was a new virus, right?
Well, the Malaysian government has a kind of quite a good report. And in that report, at one stage in December, for instance, both the Department of Agriculture and the Department of Health both went to the cabinet and said, this is Japanese encephalitis.
Yeah.
Even though pigs were dying and humans were dying and people had been vaccinated and so on.
Right.
So there was a lot of misinformation, I suspect.
And these are the kinds of confusions that now we try and avoid by being ready, right?
Yeah. One thing I may add, this is like before the next-gen sequencing days, right? So I always say, one day we hope to have a movie, how Nipah was discovered, because John played a very important role there because he knows both Ken Lam and Duane Gubler very well.
Yeah.
So he was involved in the Hendra story. So this kind of, half is a science, half is a personal context, and made the discovery possible. Otherwise, I think we'll be in the dark for a long time, and more people will die,
Didn't they make the movie Contagion?
Yeah, but...
and I have you say, Ian, you mentioned Ian Lipkin earlier.
Yeah.
Ian features in that.
Yeah.
Very briefly.
Yeah, he was the script consultant, right?
Yeah, but he's actually on the film too.
Yeah, there's a little cameo of him looking at something, But in March of 99, what were you doing?
Oh, I was in Australian Animal Health Laboratory, so I was, a part of the Hendra team, so,
Nipah was already one of the crucial people when we discovered it was actually a new virus and sequencing it and so on. So he was...
You're talking about Hendra?
Hendra, yeah. But so basically, a lot of what we know about Nipah came from the initial studies that Nipah did in Geelong.
So were you hearing about this new virus when you're in Australia in the 99?
Yeah, I heard it later. I mean, when John was talking to Duane Gubler and Ken Lam, of course, we were not involved. Until CDC, so Fort Collins, they could not really prove what this new agent is because they use all their serum as still negative. And then they linked with Brown Mahi and Bellini in CDC Atlanta. So trial carried the virus to CDC. And then finally, John suggested they should try the Hendra reagent. But at that time, the Hendra reagent was only in Atlanta CDC, not in Fort Collins.
Right.
And after they put the antibody onto this new virus, and I got the description said it's like a firework. The immunofluorescence was so strong. And so they say it's definitely Hendra-like. But Trout doesn't like the Hendra-like name, and eventually he wanted to have his own name as a Nipah virus.
Great. Yeah, if you read his review, he says, in the early hours of the morning.
Yeah.
Sounds like they worked all night to do this.
Yeah.
So, John, where were you in March 99?
Well, it's interesting. I wasn't directly involved at this point with Nipah virus, but there was some formative stuff happening with me. I was in Australia working on Australian bat virus and starting to learn how to catch large fruit bats and test them for disease. And this was a different emerging disease. This was Australia's first experience with a rabies-like virus. And I was working with Hume Field, who was actively involved in the early investigation of Nipah in Malaysia and, of course, Hendra in Australia. And he was out doing surveillance on flying foxes, large fruit bats in Australia. And at the same time, there were 3 people who had died from a rabies-like virus, and that launched an investigation of what would be called Australian bat virus that was carried by the same fruit bats and some other species as well. Okay. And so I was in Australia in 99 catching and screening bats with Hume, which would, unknown to me at the time, would launch a career in bat catching and viral testing.
Very interesting. Eun-Chung, do you remember these days?
Well, I do remember these days, but not as a Nipah person.
Okay.
So I was trying to make HIV vaccine in Boston.
Okay. Yeah. All right. So, John, why did the Malaysian-Singapore outbreak eventually stop? What was done?
Well, I think once they knew what the virus was, this was a major part of it. The fact that you could then, once you realize that the bat connection was linked to bats or the trees which were giving shade to the piggeries, that the bat urine and so on was going into the piggery, and so that connection meant once you get rid of the pigs, once you move the fruit trees away from the piggeries as well, all these things that then kind of added up to the fact that it could be controlled. It was controlled.
So the bat connection was made as a consequence of the Hendra story, is that right?
Basically, yes.
Right,
Yeah.
Okay. So the piggery was actually right under trees where bats.
A lot of the piggeries were. The farmers had two incomes. The main income was the pork. The second income was fruit.
Fruit, I see.
And the fruit trees had two were also there for shade as well.
So now this is what we call biosecurity on farms.
Right.
We keep the pigs away from. We don't even let them be outside anymore, right?
Well,
so that avoids this. Or in theory, right?
In theory.
In theory.
But just while we're on this point, it becomes a really poignant example of something that's happening all over the world now, is that we, people, are creating these opportunities for viruses to get from wildlife into domestic animals. And here's an example of, unknowingly having fruit orchards, which were a massive attractant to these fruit bats. In fact, they were considered an agricultural pest by most farmers in Malaysia, but they'd come in, decimate mango crops and other fruits just by eating them overnight. And because there were branches overhanging the piggeries, bits of fruit were being dropped in to the pigs. Right. Probably also in addition, farmers were feeding dropped fruit to their pigs that they couldn't otherwise sell. So here's opportunity for a bat virus to make its way into pigs, whereas normally bats would never have contact with pigs or other domestic animals.
Sure.
But we created this artificial interface that allowed spillover to happen.
And also the bats masticate the fruit and spit out the spats, if you like, of the fibers. They also go into the piggery.
So is that why we never saw this before? Because the pigs were never of this extent? They were smaller farms and so forth?
It only, as we started to get involved, meaning, myself, Peter Daszak, Eco Health Alliance, we started to ask some of these ecological questions and epidemiologic ones about why Nipah emerged on that index farm, on Leong Sing Nam. And the question you just asked was an important one. Why weren't we seeing this before? There were pig farms all over Malaysia. There were these fruit bats all over Malaysia, so why did it happen there? And ultimately, one of the advantages of the size of the farm where Nipah emerged is that they kept good records of their pig health. And by looking at those records, and this was work that was done by Juliet Pulliam at the time, we're able to see that the size of the farm and the intensity of the operation, the fact that so many pigs were producing piglets and that there were so many pigs populating that farm at one time, actually provided the critical mass of pigs that could be susceptible to Nipah virus to allow circulation on that farm to that extent. In other words, a smaller farm might have had Nipah introduced by a bat, but the virus would quickly move through pigs, they would largely recover, and it might go unnoticed. But a farm this size, 30,000 pigs on Leong Sing Nam, supported an extended outbreak and circulation of Nipah virus, which allowed farm workers to ultimately get sick from it, and for sick pigs to be moved off that farm over time.
And I guess now we don't raise pigs in the same way, right? We keep them away from trees.
Correct.
Is that correct in Malaysia still and here in Singapore?
Yeah, though actually the areas where these farms were, they've stopped doing piggeries altogether. It's now palm oil and so on.
Now in the Hendra story, how did the bats contaminate the horse?
Well, in some ways, it's again humans. We've been cutting down the native forest. The bats normally feed on the nectar from the flowering gums and so forth.
Right.
They had to go somewhere else. So where's best? Around humans. We have ornamental plants. We have with lots of nectar. We have fruit. So basically, they moved into areas which we hadn't normally seen bats in such numbers.
Right.
We have all the horses peripherally in the peri-urban areas where people used to have their little farmlets and have their horses. So bats and horses became much more closely linked, shall we say?
Right.
So
as far as I understand from this meeting, there are no more big outbreaks in pigs since then. Is that correct?
That I'm aware of. No, that's correct.
It's limited to Bangladesh, which we'll hear from John, and India.
Yes.
But they're
correct.
All right. And that's why this meeting is here, because this continues, right? So, John, what would you say are the main lessons learned from that initial outbreak?
I think one of the main lessons were, one, obviously, is that there's a number of viruses, new viruses in wildlife and particularly in bats, and I think that's given rise to a whole industry, if you like, of finding new viruses in bats worldwide, first of all. Secondly, that a new virus can arise anywhere, anytime, any place, unexpectedly, and cause enormous problems in terms of human health and economies of countries, potentially even globally. It shows us that we really do need to have good surveillance systems in place and be able to recognize and respond to new diseases when they arise. It's very, very important, and we do have these, that we have international and local collaboration and cooperation. You can't do it alone. So these are all lessons, if you like, which are more general.
Yeah.
In terms of the actual Nipah virus, I think we had lessons that once you know the ecology, you can very often control. In other words, as we talked about in terms of the piggeries and the shade. So these are all the major lessons I think that came from it.
Okay. Linfa, was this the first bat zoonotic infection?
Hendra, you mean?
Hendra, Nipah, yeah.
Yeah, at the BSF-4 level, but I think even more than 100 years ago, we know that rabies, for example, has a bat reservoir. I always say rabies should be the first major zoonotic disease known with a bat origin.
Okay.
But I like to think Hendra opened a new era in terms of our attention to bats as a reservoir of these highly lethal zoonotic viruses. Yeah.
Of course, we knew that viruses can come from animals, non-human animals, Lassa, for example, from mice and influenza viruses and so forth. But I talked to Peter Daszak earlier, and he agrees with you that there are more viruses in bats than other animals.
That's right.
So you said that this summer.
We debated for the 10 years of our bat special as a viral reservoir. So I was in the camp of say, yes, they're special.
Right.
And Peter Daszak was in the other camp said, no, only, you look for it and you get more. So he tried to prove I'm wrong. So he took him 8 years to build the data and 2 years to publish the Nature paper.
Right.
Eventually he proved I was right, so.
Yeah.
In my whole career, this is the only time that my competitor publishes paper in Nature, I was happier than my own paper.
But this is a good point that Linfa's raising, is that the discovery of Hendra and then Nipah did really launch a line of inquiry and research to understand, the ecology and sort of origins of this virus, but it led to other discoveries. For example, SARS. Linfa and I and many others worked together on understanding where SARS came from, and that ultimately led to bats as well, though a different type of bat. And since then, there's been so much research. The idea that bats are special as reservoirs really came about because the body of evidence was generated that indeed, diverse species of bats were carrying zoonotic viruses, and we're just starting to understand the extent of that.
So what's interesting too is you started off initially with the concept that Hendra and Nipah were in Pteropus bats.
Right.
And we thought that was it.
Right.
Now, of course, we now know they're also in Africa. And there's some evidence now they're in South America.
Yeah.
So, it's much more, the whole thing's kind of opened up a huge panoply of work that can be done.
Yeah. And they're 20% of all mammals, right?
Yeah, that's right.
Huge numbers.
Yeah, and they live very long lifespan. So they, and anecdotally, they also are more sort of prone to persistent infections. Yeah.
Right.
And, bats live everywhere people do. So we have an incredible amount of contact with bats on all 6 continents. So here's a group of animals where there's a lot of opportunity for viruses and other pathogens to jump.
Yeah. Just about every single virus family has been found in bats.
In bats, yeah.
It's incredible. And there's the enormous number of different viruses is just stunning, really.
So since we're talking about bats, John, you talked about surveillance that you do in Bangladesh. So tell us why you're doing that.
Why is to understand how Nipah operates in nature. And I think part of the idea of understanding risk to people is understanding, how, first of all, what the natural reservoir is for a virus like Nipah virus. Is it a rare disease in its host? Is it common? This informs the opportunity for spillover. It informs risk. And why we're doing it in Bangladesh is actually, it started really going back to Hendra in Australia and then in Malaysia, where Hume Field had been working on understanding Hendra virus in flying foxes. When the recognition was made, the realization that similar bats in Malaysia might be carrying Nipah, and then evidence started to mount for that, we started to survey bats in Malaysia to see about Nipah virus, to try to answer the question why it emerged on that farm in northern Malaysia. And what we found by looking all over the country was that the two species of flying fox, Pteropus bats, which are related to the ones in Australia, both carried Nipah virus, and it was pretty widespread, or at least common, to find evidence of infection in the form of antibodies against Nipah virus. But the infection itself was rare. We learned that in Malaysia, that it wasn't that common for bats to actually be infected. It was about 1% of the bats that were actively shedding at any time, which is low, but there are hundreds of thousands of bats, around the country in different colonies. So we needed to understand, how frequently bats might be transmitting virus, where it could infect pigs or even people. Then we took the lessons learned from Malaysia and brought them to Bangladesh to really help answer the question there, why are we seeing the pattern of human outbreaks that we are? And at the time, this was in 2006, and we heard Steve Luby talk about an early recognition that date palm sap was contributing to human infection. And the question was, there was a pattern of infection that was starting to show that we didn't see in Malaysia because there was just one outbreak, but we were starting to see annual cases in Bangladesh that were clustering within a time frame, winter months between November and April, and in a certain part of the country. And the question was, why? So date palm sap started to become a really clear reason for that there was a seasonality to the harvest of date palm sap. There's a species of bat that's common throughout the Indian subcontinent, Pteropus medius, which is related to the bats in Malaysia and Australia. And it's the only large fruit bat, large flying fox, in Bangladesh and India. So the connection again was made there that this was likely a carrier for Nipah. We saw that these bats were feeding on date palm sap, and that was the opportunity for spillover. Again, a human-provided interface, but in this case, people were getting directly infected rather than via pigs or other animals. And so why do the ecological studies was to try to answer the question, is there a reason for the pattern of human infections that has to do with the viral dynamics in bats? Are bats more likely to shed in the western part of Bangladesh? Are bats in other parts of Bangladesh infected? How come we're not seeing cases there? What's the story? So we started exploring the relationship between people and bats in Bangladesh, how bats are using date palm sap, how common an infection the virus is there, and also what is the genetics of the virus. What's the diversity of Nipah viruses being carried by these bats? And does that have something to do with what we're seeing in people? So we took a lot of lessons learned from work in Australia and Malaysia, all the same collaborators, and we set to work in Bangladesh to try to answer these questions.
So you've been doing a long-term 6-year study, right?
Right.
I remember from your talk. Now, how do you sample? Do you have to catch the bats or?
Yeah, it's challenging, and every environment and country is different in terms of how we do it. But essentially, these bats roost in trees.
Right.
Sometimes they're in very hard to get places, like in Malaysia, the bats were roosting in the middle of mangrove forests in tidal ocean water. In Bangladesh, thankfully, it was a lot easier, where you tended to find bats in villages, by the roadside, very common, and a lot of close association. So you set up large nets, which effectively are the size of, let's say, an area, maybe half of a tennis court, and we hang them up like a shower curtain, 2 large masts that are mounted in the treetops, a line running between those masts and the net hanging down. And we position this in front of the bat roost so that when they leave the roost in the evening or when they're coming back overnight, they fly into the net.
Okay.
And we're there waiting, and we pull the bats out one by one, very carefully and humanely. All of the surveillance work we're doing, we're very conscious of not harming these bats. They're incredibly important environmentally. These are pollinators and seed dispersers. So we try to be as minimally disruptive as possible, but we catch these bats, maybe 20 in a night. We set up a mobile lab kind of roadside right by the colony, and we'll, in the case of these large fruit bats, we'll anesthetize them. We'll collect throat swabs, urine, feces. We'll take some blood, do all kinds of measurements, and then we recover the bats and let them go. Just after that. And this gives us information really about routes of excretion. We're interested in how Nipah is getting out of bats and into people. So that explains the types of samples we collect to see how it's shed.
What did you say, about 1% of any colony is positive by serological tests, right?
That's by virological tests. So the 2 main types of screening we do are serological, looking for antibodies. And in that case, anywhere from 15 to 60% of bats have antibodies against Nipah, almost everywhere we look. But actually detecting virus, which we do by PCR, that's about anywhere from 0 to 3%. So often we get no bats that are positive. The most we ever got was 3 out of 100.
So in any given tree, with 100 bats, maybe one of those might be positive, essentially.
Yeah, but
right?
the other thing we learned and the need for the long 6-year study was that there is a periodicity to an infection. And depending on the timing of when you sample, you could find nothing or you could find more virus. And so we needed to understand if there were seasonal patterns to that. And it was only by doing consistent, long-term, repeated sampling that we started to get an idea that, yeah, there are actually epidemics that happen within these bats. And they're not necessarily annual, but they are periodic.
So you want to understand the drivers so that you can be prepared and say, okay, this part of the year is not good for you to get near bats or whatever, right? That's the idea.
Well, yeah, and I think what we've learned from Bangladesh is there's a real important balance between human behavior and the timing of, say, cultivation and harvest of date palm sap that leads to outbreaks, and what's happening with the bats and the epidemiology within the animal. So whereas bats are capable of shedding virus at any time of year, and depending on the where and the when, it might influence the timing of shedding, the interface, the date palm sap, becomes really critical in Bangladesh for spillover. And then when Kerala, India happened, it's a different story because their date palm sap isn't part of the equation, and we still don't really know yet how the first case was infected.
Right. So you and many people say that the infection does not harm the bat.
That's right.
So how do you know that? Because you get PCR positive animals and they seem fine?
Yeah, well, there's kind of two ways we know that. And one way is that in the field, we see bats that are infected that we later test positive that are apparently healthy. So they look like every other bat, they're flying around happy. But really the other way to answer that question is through experimental work.
Experiment challenge, yeah. So collaborate with John and Peter, others. We have import bats from Malaysia and go to the Australian Animal Health Laboratory in Geelong, and we did a challenge studies. So in theory, the Australian bats have never seen Nipah virus. They have seen Hendra, right?
Right.
But we have done this for Ebola. We have done this for SARS. So I think that's commonly accepted that bats of different species, even if they have never been exposed to that particular virus, when you challenge them, they don't develop any symptoms, not even fever. Yeah.
And that's a goal of your research, to understand why
That's right.
In fact, we covered your recent paper on
That's right.
on TWiV to understand that as well. Now, John, you mentioned in your talk that you think the virus went into Bangladesh bats in 1941.
Yeah, and take that with a grain of salt. We looked at the available sequences that are out there for Nipah virus from bats, and to be honest, it's a good number, but it's not a huge number. And so what we know based on mutations in the virus lets us sort of infer the movement of that virus. And so, yeah, the data that we have now suggests that it was introduced maybe sometime in the 1940s from India into Bangladesh. So that's not saying anything about maybe how it moved from Southeast Asia. And at that point, it radiated outwards within Bangladesh via bats and then has made its way back. But again, that will change as we get more and more viral sequences to inform that analysis.
But surely the virus is much older, right?
I think so, yes.
Much older.
Bats are millions of years old. They're an ancient order of mammals. And so, I think a lot of the assumptions we make is that they've co-evolved with these viruses. And the evidence for that is, one, that they tolerate the viral infection so well. And also, Nipah viruses are pervasive pretty much everywhere we see Pteropus bats, which extends from Australia throughout Asia, all the way to Madagascar. There's evidence for Nipah virus infection.
In some bats, there are inserted viral DNAs in the genome, so you can date the point of existence. Does that exist for Nipah at all? Do you know? Are there pieces of random viral DNAs in the genome?
The EVEs, they call the endogenous viral elements, I mean, from the publicly available genome sequencing in bats, we haven't found the Nipah virus. We have found lots of other viruses, like the retrovirus, for sure,
But they aren't all virulent. There are some Nipah viruses which are totally avirulent as far as we can tell. There's one called Cedar, for instance.
That's right.
In Australia, which is not associated with disease at all.
Right.
So there's quite a broad range.
Sure. Not surprising. So I guess
the other point in terms of virology, for Nipah virus, when we first sequenced the Hendra genome, we're very surprised. In the family of Paramyxoviridae, the genomes are very stable. So it goes from 15.2 to 16 kbs. No virus have gone, shorter or longer genome, and Hendra is at 18 kilobase. So that's a substantial increase of the genome size. To this day, we still don't understand these untranslated regions, extra bits of RNA in Hendra, what they're doing, And secondly, is that an old version of paramyxovirus or is a new version of paramyxovirus? So that still remains to be answered.
Yeah, yeah. We heard at this meeting over and over, there are no good rapid diagnostics. And so the question is, are there spillovers occurring into people a lot and we just don't see them? Because it sounds like there's some asymptomatic infections, right?
I think for sure there are spillovers that we're not noticing, and some of that is a product of just lack of looking, so poor surveillance, not just diagnostics, but not even having infrastructure to recognize or diagnose cases of encephalitis. And there are so many different cultures and groups of people that interact with bats in different ways. Bats that we either know or assume could be carrying Nipah viruses, and we just haven't looked yet to see whether they're being exposed. But I think there are new tools available now. Some of them are serologic, looking at antibodies and exposure that can start to fill in the gaps in terms of who's getting exposed and which populations are vulnerable.
So John, you mentioned that there are related viruses in different bats in other parts of the world, and we heard in South Africa, right? They're fruit bats, Egyptian fruit bats, right? Yeah. And so that's scary, right? Because they're related, they're similar viruses, they could cause human disease as well.
It could, but it hasn't been found yet in Africa. But almost certainly, I suspect, once surveillance gets better and so on, they'll start appearing.
But I guess, like John Ivers sort of collaborate in the Hendra, but also in the coronavirus, right?
Yeah.
So personally, I always found that, the SARS, was a big kind of a story that we trace back to bats. And then, in the bats that we found the SARS-related virus. So most of the international attention was going for that SARS-related virus and say where they are and are they ready to jump. And then we had a MERS. Right. First, it's not in the hot zone, the traditional hot zone of Africa and Southeast Asia. And secondly, on the tree is quite distant from SARS. So I always say, in my lectures, that if SARS can jump and kill people, MERS can jump and kill people. In between, there's hundreds of other coronaviruses. So that's same for Nipah virus. We have lots of Nipah-related virus, and the classification is difficult. But I think classification is the last thing I worry about because it's all academic, joint line, right? Virus evolves continuously.
Yeah. And MERS is an interesting example because the MERS investigation started with bats because of the relationship with SARS. And I was involved, this was with Ian Lipkin too, going to Saudi Arabia and trying to identify the origin. However, the more we learned that even if MERS comes from bats originally, camels are really the most important host in terms of human infection. And it's only when you start to look at the ecology and the sort of relationship between reservoirs and people that you start to piece these parts of the puzzle together.
Yeah.
And even more for that too is that it must be quite a recent virus in camels. But if you look at Australia, where we have the largest number of wild camels are in Australia. And they're having been brought in from in the 1880s. We found no evidence of MERS in those camels.
No evidence?
No. So it suggests that it must be quite a recent virus.
And they came from where?
They came from different parts of, some came from around Afghanistan, some came from North Africa.
It's interesting, it's recent. Well, camels are interesting because the smallpox is thought to have originated in camels, maybe through a gerbil or something like that. But they moved quite long distances. I think they originated in the Americas and moved over the land bridge and ended up in the Middle East and so forth, and Africa. So they're quite interesting. Are there any Nipahs in bats in North and South America that we know of?
There's been some evidence, I believe, in South America. And I don't know much about
no sequence yet.
Yeah, it's serologic evidence of antibody, but there's been no real description of a Nipah virus.
So you go to places and survey where there's a clear threat, but there are places where there are no threats, but maybe should be surveyed also, right?
Yes.
Is anyone doing that?
In fact, we just now are at the tail end of a massive 10-year global project that was funded by the U.S. Agency for International Development called PREDICT. And it was for just that reason, recognizing that bats and other groups of animals, including rodents and non-human primates, are important reservoirs for zoonotic viruses. And identifying parts of the world that are particularly vulnerable to emerging diseases based on the diversity of animals that are there and the way that people and animals interact, we set about starting to look for novel viruses and characterize high-risk interfaces, ways that people and animals are interacting to try to head off disease X or the next virus from emerging.
So, John, before I leave you, you have a paper being reviewed now. Hopefully it will get accepted and we can chat about it. I always say, you know, you and I meet everywhere but where we live in New York.
That's right.
So we met in Washington and now here, and maybe we can meet in New York.
We should try that sometime. That'd be nice. I hear they have good coffee.
Yeah, it's not bad. Eunchung, you're going to talk after this about vaccines.
Right.
And I've seen many posters and upcoming talks on Nipah virus vaccines. So let's talk a little bit about that. For whom do we need a Nipah virus vaccine?
So I can think about at least two scenarios where...
Did I say Zika? I said Nipah.
You said
You said Nipah.
Sorry.
Yeah. Well, we can talk about
Jet lag.
So, you know, the high-risk areas where like the Nipah belt type, and as you said, there are a lot of bats having positivity of Nipah, so it can happen anywhere. And of course, secondly, outbreak response. You can treat the healthcare workers or the people who, you know, surrounding the index
In
cases.
a place like Bangladesh, where there are rather regular cases, that would be an area where you would just immunize people who are at risk. And I think your surveillance would be part of saying who gets immunized, right?
Absolutely, is figuring out who's on the front lines dealing with cases. And,
Yeah, so there will be a I mean, of course, they have to be willing to be vaccinated, but there will be a candidate.
I can think of third category is the scientists who are researching this field.
Exactly.
Yes, of course.
Yeah.
No, seriously, if you have a
Scientists,
vaccine today, many people will take that, you know.
Yeah.
So
You don't need to be a cell phone.
yeah, that's right.
And healthcare workers.
And in Australia, for example, the vets, the equine vets are really crying out for a vaccine because as a OHS sort of vaccine, they would take it.
One health solution.
That's right.
Yeah. And also, if you have an outbreak, then you go in and you immunize, you do ring vaccination, I guess.
Yeah.
So there is a need.
But on the other hand, in Australia, we do have a vaccine for horses.
Right.
And it's just not taken up to a large extent. It's too expensive. But there's always a risk now for any vet going out to look at a sick horse. And,
I thought horses are expensive too.
yes, absolutely, yeah.
The racing horses
racehorses,
but not the hobby horses in the backyard of your house. Yeah.
So the vaccine is good, providing it's used.
That's right.
I guess.
So, Linfa, you were involved in the development of the Hendra vaccine, right?
Yeah.
At Geelong, right?
Yeah. With Chris Brodel from USA and funded by NIH. Yeah.
Really?
Yeah, of course.
Of course.
Of course, of course.
I don't know if of course. So what kinds of vaccines are being developed?
So I think Stephen Ruby talked about the other day, like there are very many good vaccines. There are different platforms using subunit vaccines, or at least 9 different kind of viral vector vaccines. Now the nucleic acid vaccines, including messenger RNA vaccines. So there are a lot of very good, effective vaccines being tested in animal models.
So like a VSV is being used as a backbone for some.
Yeah, VSV with Ebola, for example, one of them. And so it may be able to cover both.
And Linfa, you mentioned that the Hendra vaccine will protect against Nipah.
Yeah.
That's remarkable, right?
Yeah. Better than Hendra. I don't know, understand. Scientifically, we still don't understand, but the soluble G, initially, for political reasons, is funded by NIH and partially by Australian government. So we had to go with Hendra as our first priority, and Chris agreed. So we put a soluble G of Hendra, into the ferrets, into horses try. And when we prove it's working, then we shift to non-human primates, and then started challenging, the Hendra, the Nipah Bangladesh, and Nipah Malaysia.
Right.
And the solid G from Hendra seems to be the best.
Interesting.
Yeah, so
very interesting.
And it's proven, and so I think this is the one is going to be reformulated as a human vaccine. Yeah.
And I think broad spectrum coverage is really important when we start to talk about these emerging viruses, because we have to get away from the idea that it's just one pathogen, but we're really talking about a spectrum of viruses that are correlated that might have slightly different clinical profiles in people, but essentially cause the same disease. And we need a vaccine that's going to protect against the range of related viruses.
Do you think people will be better than horses at taking this vaccine?
I think so. Again, we still have this big problem: who's going to buy it? Who's going to pay for it?
Well,
that's the kind of, to me,
in the areas where people need it, they're not going to buy it. You have to give it to them.
Yeah, that's correct. Right? That's correct.
So just like with the Hendra, NIH is also interested in a Nipah vaccine as well.
Definitely, yeah. So I think, as Nipah pointed out, most of the vaccines developed, funded by NIH.
So from what I could hear at this meeting, there are two issues. One is, it seems like viruses from Malaysia and Bangladesh are phenotypically different. So which one do you use, or do you use both in a vaccine?
Well, Nipah may disagree, but...
Hendra is protecting all the different...
So you just use Hendra?
I think for the current virus, I think the Hendra virus, the Nipah Bangladesh, the Nipah Malaysia, even with their sort of mutation rate, and we have done long studies, this virus is relatively stable. So for these three viruses, I'm confident to say the vaccine will protect.
Okay.
But if you have a Hannibal-related virus from Africa, from China, jump, and they also cause disease, then I'm not so certain that they will protect. But I think in this afternoon, we're going to hear this, the CEPI is also funding this platform base, Paul is in the audience, so they try to have a platform ready that you just switch the envelope protein, and hopefully, we'll hear this afternoon. They're very ambitious from gene to vaccine in 16 weeks. If we have that, then, Hannibal-related virus will be covered very easily.
The other issue is which animals do you test it in for preclinical, right? And we've heard a couple of different animal models. Do you have any thoughts about that?
I think all three models are pretty good. They're, ferrets, African green monkeys, and hamsters. They all have symptoms similar to human disease, even though there are some slightly differences. And I think most of the vaccines evaluated in these models showing all protection. So I don't think that in terms of the evaluating vaccines, their difference is pretty small. I think maybe therapeutics may be slightly different matter, but for vaccine evaluation, I don't think there's much difference between.
Okay, so a number of candidates have shown protection in animal models, and they're ready for phase 1.
I think one of the vaccine is going to.
Okay, so phase 1 is easy because
the safety study.
Just safety. The real key is efficacy.
Right.
So how do we do that?
I think the just previous session covered this the accelerated approval program. So if you want to get the licensure in US, there are two options. One is going through that. So that is using biomarkers that predict clinical outcome as an endpoint. So then they will facilitate the licensure, but it has to be proved by later clinical trials and have to get really approved just similarly to double-blind study. But that is one of the methods you can do, and we had a whole discussion about that. And
that's because we don't have big enough outbreaks to do a real, powered clinical trial, right?
Right, exactly. Yeah.
I mean, that was the case with Ebola until we had a big
until it wasn't the outbreak.
Western African outbreak.
Hopefully it doesn't happen with Nipah.
No.
But
One good news about the Nipah vaccine, I'm personally kind of optimistic, is the correlation, the efficacy of the vaccine in all the animal models against all the viruses, that the neutralization, the serum neutralization is pretty good correlate. So so Hannibal, I think the T cell immunity is almost neglected because one is difficult to do. Secondly is that the antibody titer neutralization correlates so well with protection in an animal challenge experiment. So we are, quite quietly optimistic to say when we go to phase 1 human clinical trials, if they induce sufficient neutralizing antibodies, then we are pretty confident, in an outbreak situation, and as long as they're safe, I think the chance of us succeed is very high. Yeah.
So it's interesting to see the way the approval of biologicals is slowly changing, and the polio really started this because China and Japan have made Sabin IPV without an efficacy trial, but just looking at antibodies and saying same profile that you get, and we have to go by that. And
the other is you're dealing with a life and death disease, right? So in Australia, we had a family. Unfortunately, a mother and a daughter was exposed to a dying horse, and eventually proved that died of Hendra.
Right.
And then Chris Brodo, my collaborator in USA, has this monoclonal antibody. So he will present, late afternoon. So this is proven to be very effective in our ferrets model. So we offered to them to say, we have this, but it's experimental medicine, basically has no approval in terms of clinical use. And then we fast-tracked to the equivalent FDA in Australia. Within 48 hours, we got approval. We fly the antibody from DC to San Francisco, Sydney to Brisbane, and we apply. So personally, I feel like if we have a vaccine of Hendra or Nipah, if you have the safety done and you have the phase 1, phase 2, and with very good at neutralizing antibody, then I think during outbreaks, the threshold to overcome to use will be relatively low. Yeah.
So that's one of the areas we didn't talk about, but had sessions at this meeting about therapeutics. And
so it's coming
really,
this afternoon.
This afternoon, monoclonals, small molecule inhibitors. It's really remarkable the progress. We also didn't talk about diagnostics, but there were sessions on that. Transmission. I think there was just something about what you need for clinical trials just recently, right? The previous session. So it's really remarkable for one virus that there's all this activity, right?
Yeah, I'm biased. I always say, in the emerging zoonotic virus, maybe the Nipah virus the community is very united. We collaborate so much from discovery of the first outbreak. Even the reservoir, was sort of sorted out within a very short period. And then we had animal models, we have vaccines, therapeutics, monoclonal antibodies, all within 20 years, basically.
And that's the model for any new emerging. That's disease X on the WHO roadmap, right?
But we do need a really, very transparent and close international collaborations.
And multidisciplinary. And I just want to add that Nipah's really been, to me, it's storybook in terms of illustrating the need for a One Health approach. And the government of Bangladesh has particularly done a good job embracing the idea of doing surveillance in wildlife contemporaneously with outbreak investigations. So while they are investigating an outbreak in people, there are people on the ground screening bats to see what viral strains are circulating locally, which informs the understanding of what's getting into people. And we didn't used to see that very much with other diseases. So here we have a broad community scientifically that's added those puzzle pieces to understand this, and now it's being applied epidemiologically in terms of public health, which is kind of the ultimate goal, right, is to protect lives.
Well, also this illustrates different people involved, not just governments and industry, but you have nonprofits like yours and others that are involved, and many, many more countries than we ever saw before, which is really key to establishing capabilities elsewhere, right? So tell me, each one of you, tell me one thing you learned from this meeting that you didn't know before. John?
I guess what I learned more today was about human-to-human transmission and the incidents and so on. I knew some of it, but it was put very nicely, very clearly today.
So the extent of human-to-human transmission.
In Bangladesh, India, and in Philippines, particularly.
So in the Malaysian outbreak, 99, was there clear human-to-human?
Not clear, but there was some evidence. But it wasn't very clearly.
Okay. How about you, John? What did you learn?
Yeah, I don't have as much exposure to the vaccine development side, and so it's been really interesting to hear about the challenges of trialing a vaccine in a population with a rare disease where you really have a small number of people affected, and just how extensively and how long it will take to understand efficacy.
I agree.
So that's been interesting.
Yeah.
You see the poster where they predict you need millions of doses and hundreds of years under certain
Yeah, 500 years to finally get a handle on this. And
That's not good.
you never want to hope for a situation like Ebola, West Africa, but at least you understand that when you have that kind of massive scale outbreak, it's a good opportunity to trial vaccines.
For sure. Unchung, what did you learn?
Well, actually, I learned the more scary thing that actually bats carry TSV, which I didn't know about. That's a pretty scary disease, and Harlan virus and so on. So I think, there are maybe a lot of different viruses. You may know of these, but I didn't know that, and it seems to be very scary. I thought it was tick-borne virus, but.
All roads seem to lead to bats somehow, unfortunately for bats.
You probably knew everything, right?
No, not really. Not really. Actually, the thing I think, Wanda's talk from the South African surveillance, because, as a person who has been working in this field for more than 20 years, the thing I'm most worried about is this Hannibal-related virus widespread. So she has proven that in African bats, there are many more viruses to watch out. So I thought that was, I knew her work, but the detailed science was impressive.
I also like that. I didn't know that, and I found that interesting and kind of scary. To be clear. Okay, that is a special episode of TWiV here at Nipah at 20. It's funny. I don't like to say the anniversary because the anniversary is usually a good thing, right? It's the 20th. A year of discovery or something like that.
For scientists who discover is their achievements, it's a good thing.
Yeah, for sure. And this year is also the 50th year of Lassa virus discovery. And I said earlier that there's a great book on Lassa discovery called Fever by John Fuller.
Yeah.
And I read that in the early 70s, and that's what made me want to be a virologist. It's that book. I read this is the coolest thing in the world.
That was.
Anyway, great meeting. Thank you for having me. That's you can find TWiV on any podcast player. Our website is microbe.tv/TWiV. You can send your questions and comments to twiv@microbe.tv. And if you like what we do, consider supporting us. Financially, go to microbe.tv/contribute. My guest today from Duke NUS Medical School, Lin-Fa Wang. Thank you so much. And thank you for inviting me.
Yeah, I'm glad I finally got you after 3 trials,
We're generating a lot of content here, and I'm visiting your school tomorrow and more on Thursday. Thank you very much. From Curtin University, John McKenzie, thank you very much.
My pleasure.
I like the historical. I think you can't forget history, right?
No, absolutely not.
Otherwise you repeat it. From Eco Health Alliance in New York City, of all places, John Epstein.
Thank you, Vincent, and look forward to seeing you back in New York City.
Well, when your paper is published, come up and we'll discuss it because there's a lot of points that we can go over in detail. That would be fun.
Thank you.
And from the NIAID, Unchung Park, thank you so much.
Thank you. Your pronunciation is great, improved.
You could thank Ian for that. I used to mispronounce it all the time.
Yeah, you should thank Ian.
I want to thank ASV and ASM for their support of TWiV. The music you hear is by Ronald Jenkins. You've been listening. Oh, and I want to thank the audience. Thank you for coming. Appreciate it. There are like 50 dedicated people standing here. We appreciate it very much. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.
Thank you.